Dyskeratosis congenita is an inherited bone marrow failure syndrome that's characterized by increased cancer risk, but it can be very challenging to diagnose. Patients with DC have a very wide range of problems that can present at any age and seem to vary within families and between families. There are many genes that, if they're mutated, can cause DC in our patients. And all of those genes are related to telomere biology. And it's telomere biology that we think is causing the problems with DC patients. Patients with DC have extremely short telomeres, and telomeres are the ends of our chromosomes. They are responsible for keeping our chromosomes stable, but they shorten with each cell division. And with successive cell divisions, telomeres get shorter and shorter, and eventually cells undergo apoptosis, or they die. And so telomere length is really sort of a marker of biological aging. Patients with DC have very short telomeres. This was first figured out when the first gene for Dyskeratosis congenita was cloned in 1999, and it's called Dyskerin, or DKC1, and those studies led to the first findings that telomere length was abnormally short in the cell lines from those patients. Patients with DC generally have telomere lengths less than the first percentile for age, so that means they're extremely short. There are such differences in the clinical severity. So there are the patients who have the Hoyle-Riederson, or HH variant of DC, who tend to present when they're very, very young as babies or toddlers. They have cerebellar hypoplasia, a small cerebellum, and immunodeficiencies, plus problems associated with DC, like the nails and skin changes, the gastrointestinal changes. And they're little children who have a lot of medical problems. Another related variant is Revesz syndrome, which has features of DC, but also has exudative retinopathy, or some bleeding in the back of the eye. And then we have patients at the other end of the spectrum who are older adults who have a mutation in a gene that has fewer clinical features, or has features that don't develop until they're 40, 50, even 60 years of age, tend to have the complications not present themselves until adulthood, tend to have mutations in genes that are involved in the telomerase complex, in TERT and TERC, and some other related genes. Whereas the more severe patients with the Hoyle-Riederson variant or Revesz syndrome tend to have mutations in DKC1, in TIN2, in RTEL1, in TPP1.